I guess you'll be still.
That's true. That's true. Who wants the front row?
Do you want to ask people to come get seated? Yeah.
Who's sneaking out early from this?
Everyone just go.
I mean, it's not like it's the Four Seasons. It's like some sandwiches.
Ethan. 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 You're up.
Okay. All right, good afternoon, everybody. Before I introduce myself and today's briefing topic, please allow me to begin with acknowledgments and gratitude. First, thank you to the EveryLife Foundation for giving me the distinct honor of moderating today's Congressional Rare Disease Caucus briefing. I will do my best to amplify the voices of patients suffering from the invisible plague of 10,000 plus rare diseases that has shrouded not only the lives of over 30 million Americans, but also their families and local communities. 99% of all rare diseases are what can be considered ultra-rare or small rare disease populations. Next, I would like to thank the members of the Congressional Rare Disease Caucus and their staffs for support, dedication, and camaraderie in this righteous battle against rare diseases. Senator Amy Klobuchar from Minnesota, Senator Roger Wicker from Mississippi, from my home state of California, Representative Doris Matsui, and from my birth state of Florida, Representative Gus Bilirakis. Finally, on behalf of everyone today, I would like to thank the following rare disease community sponsors. Let's see. Sorry, these are the Rare Disease Caucus members. And then I would like to briefly thank Alexion, Amgen, Biogen, CS Behring, Genentech, and Sanofi. Okay. My name is Ethan Perlstein. I reside in Oakland, California. I'm a scientist, biotech entrepreneur, and investor. I'm also the father of two beautiful boys, Kai, 9, and Luca, 4. My younger son, Luca, has an autism spectrum disorder diagnosis. I discovered my life's purpose a decade ago in 2014 when I became founder and CEO of Perlara PBC, the world's first biotech public benefit corporation created to partner with rare disease families in search of treatments and cures. I'm also the co-founder and CEO of Maggie's Pearl, a joint venture between a rare disease family from Sturgis, Michigan, the district of former U.S. Representative Fred Upton, I should add. Joint venture between that family, Perlara, and the Mayo Clinic. I will return to Maggie's Pearl at the end of my remarks after I frame up the topic of today's briefing, which is novel approaches to drug development for small rare disease populations and the policies that can drive them. I've spent the last decade toiling in the trenches with truly remarkable and unstoppable families whose children were struck by genetic lightning. Oops. Based on those indelible experiences, I believe the future of ultra-rare drug development will be family-driven because no one else cares more about this problem than those who are personally ravaged by it. But families won't be able to go it alone. The august panel of experts convened for today's briefing represents the major stakeholders in ultra-rare drug development. Families and patient communities, of course, and both government and industry partners. In a few moments, they will share each of their experiences and proffer policy perspectives and constructive solutions. 40 years ago, the Orphan Drug Act codified that a rare/orphan disease affects fewer than 200,000 people. It created a patch quilt of incentives for medicine makers to meet the treatment needs of these smaller communities through tax credits, fee waivers, and a potential 7 years of market exclusivity after approval. Unfortunately, these policies are not enough for the long tail of diseases, which affect many fewer than 200,000 Americans. In reality, it's as small as tens, up to hundreds, but no more than a few thousand total patients affected in the US by the average ultra-rare disease. Everyone's definition varies, But there are many shared challenges regardless of the exact numerical threshold used to define an ultra-disease population. Take, for example, 2,000 patients in the U.S., a reasonable cutoff suggested by ultra-rare drug developer extraordinaire Dr. Emil Caucus of Ultragenics in a proposal he dubs the Ultra-Orphan Drug Act. There are successes, too. Treatments for tiny populations have made it across the finish line, but it has taken Herculean efforts from the patient and families affected, and the right formula of federal investments in basic and translational science, innovation in how we execute clinical development, and successful application of regulatory flexibility, among other factors. Now, meet Maggie, a medical pioneer. Maggie is from Michigan and has PMM2-CDG, the most common congenital disorder of glycosylation, an inherited metabolic disease that affects over 1,000 patients globally, most of them kids like her. 4.5 years ago, Maggie started taking a repurposed drug called Apaurestat. A pivotal phase 3 trial is now fully enrolled and expecting readouts later this year. Here's a montage of Maggie's historic N of 1 study. I think the video speaks for itself. Sorry for the cheesy background music. But this is Maggie's baseline about 7 months before she started taking this medicine. So this is what walking was like for her. This is at the hotel. Anyone ever been to the Mayo Clinic? This is the Grand Collar Hotel. This is the hallway. Maybe you recognize the carpeting. But this is Maggie's sort of baseline. You can see she really can't hold a straight line. And we'll just give you a sense to acclimate here. You know what? This music's a little cheesy. I'm gonna get rid of it. So, I'm not sure who's a doctor here, who has a family member with a rare disease, but maybe this is a typical picture. So she's kind of struggling here, but she is extremely, extremely persistent. I'll just give you a sense to really acclimate to her baseline. And in the next sequence, you'll see 3 months on the drug. Anyone notice a difference? Raise your hand. All right. So this is a couple months on the drug. This is back at our Mayo Clinic visit. And then this will give you another sense here. Here's a straightaway. This is a floor here where she could really start cruising. This is 6 months on the drug. And then we'll just kind of cut to bottom here, which is, oops. Can I not advance this? Apologies here. Sorry, I don't want to make you watch that all over again, but is there any way to fast forward this? Maybe I can. Yeah, it's not letting me advance. I have to, I want to get you to the good part at the end where she's standing and walking. You know what, let me show you actually a second patient. So since one good turn deserves another, a second family caught wind of Maggie's progress because her mom, Holly, was blogging about it in real time. Word spread surprisingly fast in rare disease communities. That led to a second N of 1 study with a little boy named Ethan. This one will hopefully work out a little bit better. So here's 3 months on the drug. These are his first independent steps. Here's 4 months on the drug. And here we go, 2 years on Apaurestat. You notice he's obviously grown a lot and still able to maintain his balance in his new body. Okay, sorry I couldn't show you Maggie's, but she did something very similar. I can show you later. Okay, Maggie's pill is not yet across the finish line, but our pivotal phase 3 is hurtling down the track as fast and safely as possible. The goal of Maggie's Pearl is to prove that a bottom-up, ultra-lean, ultra-fast, and ultra-persistent family-driven model to drug development is poised to scale. It's a fully remote, family-owned, clinical-stage biotech company. By families, for families. Imagine a balanced biopharma ecosystem teeming with profitable minnows and not dominated by a few lumbering whales, all chasing the largest market. Now, please allow me to introduce our distinguished panelists. First up, Christine Wagoner. Christine founded the Cure GM1 Foundation in April 2015 in honor of her daughter Iris and all those affected by GM1 gangliosidosis. Christine's work at Cure GM1 has involved a broad range of research endeavors, including animal models, biomarkers, gene therapy, enzyme replacement therapy, patient registries, patient-reported outcomes, newborn screening, and the first ever GM caregiver preferences study. Christine?
Thank you so much for the invitation. It's truly a pleasure to be here. I flew in last night from the San Francisco Bay Area. I also wanted to point out that you all are in a room with a child who has GM1 gangliosidosis. So little Aiden over there has late infantile GM1. For GM1 gangliosidosis, a lysosomal storage disease, it is ultra rare. There are an estimated 1,000 people living with GM1 in the U.S. Like the vast majority of rare and ultra-rare diseases, there is no FDA-approved treatment. There is one small molecule that is on the market off-label that sometimes doctors try to recommend, and families try it because it's the only thing that anyone is offering, and they struggle greatly due to the cost and because it's an off-label prescription. So the majority of people don't end up getting it or trying it. We managed to try it, and to be honest, it doesn't work very well, at least not in our case. So what are we left with? We are left with supportive care, occupational therapy, physical therapy, palliative care, and hospice care. The majority of patients with GM1 are children, babies, who face certain death. My daughter Iris was diagnosed with juvenile GM1 gangliosidosis at age 5.5. Her diagnosis was in August of 2013. So I have been advocating for a treatment now for over 10 years. In this picture, you can see in the upper corner in the blue dress that actually people didn't believe her diagnosis when she was first diagnosed, and the school didn't even want to offer us services, but she has a highly degenerative neurological disease. You can see the past 10 years have taken a great toll. She has lost virtually everything. Her ability to speak, to walk, to move. She suffers from seizures. She has a feeding tube. I want to take you back. I had no prior fundraising experience, but I knew I wanted to help, and we heard of research to support. This slide says 327 people supported it in 2 months. Please subtract a month, because it took me a month. I created the fundraiser, and I had to get up the courage for a whole month before I could post it. So the outpouring of support was incredible and truly extraordinary. The initial goal of our fundraiser was originally $50,000. And I had to keep increasing the goal, and I felt I was doing it very tentatively. And we started approaching $100,000 in donations. That night, though, I went on the internet and I contacted all the Bay Area news stations about our story to ask them to give us coverage. I was at work the next morning, and they did give us coverage. All the stations called us back. So I'm going to show you a video that takes you back to where we were and how we wanted to support research, because there was no one else doing it.
A rare and deadly disease could take the life of a 6-year-old girl in Berkeley if a cure is not found soon. Iris Dooley's parents tell Crown 4's Philippe Jagal that they've taken to the web to raise money to fight this disease and save children like their daughter.
Back in 2013, 6-year-old Iris Dooley was diagnosed with the rare and deadly degenerative neurological disease called GM1 gangliosidosis. It's found in just one of every 300,000 people.
It robs children of the ability to speak, to walk, to even swallow, and to move their limbs.
Last December, her parents Christine and Doug started an online fundraising campaign on the website sweetiris.org, soliciting for donations with this YouTube video, spreading word about the disease and how people can help.
Ultimately, it's your worst nightmare.
As of now, the family has raised more than $200,000. The goal is $500,000, with the funds going to help further research on the disease at Auburn University in Alabama. but they need your help.
I don't think I could ever explain to them how much it means to us.
And as Iris's condition deteriorates, her parents stress the importance of trying to reach that fundraising goal as soon as possible to give their daughter the best chance to live. In Philippe Jagal, Cron 4 News.
So I just want to tell you, in the news piece, he said we had raised $200,000. We were about to hit $100,000. That day, someone in our extended network donated another $100,000 anonymously. The news piece aired, and then a Silicon Valley CEO donated another $100,000. So we went to $300,000, which was a matter of days. All of this was happening, and people said we needed a nonprofit. So we formed the Cure GM1 Foundation, the only 501c3 entirely dedicated to GM1 gangliosidosis ever. We have done a great deal, had many lessons, and had to mature greatly and learn about the drug development process. We have an awareness day, it's coming up on May 23rd. We have an annual conference. We recently formed a natural history data sharing consortium. We conducted a caregiver preferences study. We made animal models. We also optimized and deployed the assay for newborn screening for GM1 in the largest newborn screening study in the US, Screen Plus. I have a full-time job, and I have a nonprofit, and I have a special needs child and another child, and a husband, a family, and a life. There's very little support for ultra-rare diseases like GM1. We have also engaged with the FDA. In 2022, we had the externally led patient-focused drug development meeting with FDA, which was a watershed moment. I don't think people thought there would be that many people at that meeting. There were 217 people, 33 government employees, and 20 caregivers who served as panelists. It's a large moment for us, and we continue to fight. Iris just celebrated her 16th birthday. We've done a great deal over the past 10 plus years. I want to show another video which outlines the work that we've done. There has been a relative explosion of GM1 gangliosidosis clinical trials. Our disease is over 150 years old. There were no clinical trials until after our foundation was started. Please play. Iris, can you say hello to the camera? Hi, my name is Christine Wagner, and I'm the president and co-founder of the Cure GM1 Foundation. But above all, I am Iris and Carter's mom. We wanted to talk a little bit about the Cure GM1 Foundation, the nonprofit that we put together to help find a treatment for GM1 in honor of Iris and the global community and everyone who's suffering from GM1.
Hi, I'm Doug. I'm Iris's dad. A little over 10 years ago, Iris was diagnosed with GM1 a few days before she started kindergarten. She was bright, she was incredibly charming, she had friends and playdates just like any of the other kids.
We have been fighting for new research, new clinical trials, and a lot has happened. We're so fortunate. There have been 4 clinical trials since we set up Cure GM1.
When Iris was first diagnosed, we were told there are no treatments, there are no trials, and there is no hope. All of this has been made possible through your support. not just financially, although finances are incredibly important since these programs are incredibly expensive, but also through your volunteering, your encouragement, and your belief that someday we can make this terrible, terrible disease a thing of the past. So thank you so much.
Please join us in celebrating Iris's life. There has to be meaning to this suffering, and we are so grateful for any support you can offer. Thank you so much. I want to bring you to now a very sobering slide. While we had an amazing burst of clinical trials beginning in 2019, last month, the last company that had a clinical trial discontinued the program. So we, as a community, are left knowing that there are possible treatments that actually, in the family's view, were indeed beneficial to our community, but they are inaccessible. In conclusion, I would like to outline a few ideas regarding how it might be possible to help beyond what our family can do. We need specific incentives for ultra-orphan diseases such as GM1. We, of course, need increased financial support for and specific grants for patient advocacy groups, drug development, and more research. For a disease such as GM1, I do believe that we need accelerated approval based on biomarkers. We have excellent biomarkers. Our disease is primarily infantile. We need modernization and support for newborn screenings so that actually we can get the youngest babies possible into the clinical trials. We also need data sharing incentives and enforcement of data sharing policies. I'm from California, and there is a great government initiative called the California Institute for Regenerative Medicine. They are overrun with proposals. We need more institutes like that to help support the development of treatments and genetic medicines. Thank you very much.
Thank you, Christine. Next up, Dr. Adora Indu. Dr. Indu is currently the Chief Regulatory Officer at Bridge Biopharma. Her expertise incorporates regulatory strategy, patient engagement, prescription drug promotion, and policy for U.S. international markets. Prior to joining BioMarin, Dr. Indu was Director and Head of the Division of Medical Policy Development at the U.S. Food and Drug Administration, Center for Drug Evaluation and Research, and also a Commander in the Commission Corps, U.S. Public Health Service. Dr. Indu received her doctorate in pharmacy from Howard University's College of Pharmacy and a law degree from the University of Maryland. Dr. Indu serves on the Board of Directors for Acadia Pharmaceuticals and DBV Technologies. The dais is yours.
Thank you very much for that introduction. And good afternoon, and really appreciate you providing me with the opportunity to share a few words regarding the drug development paradigm for ultra-rare diseases. I'm here today as the Chief Regulatory Officer for Bridge Biopharmaceutical, and also a former regulator with the Food and Drug Administration, and a person whose family has also been impacted by a rare disease. So let me start by saying that the statistics that we hear very often don't lie. As you can see, rare diseases actually presents a real global health crisis with dire consequences. In 2021, for the first time, the United Nations General Assembly adopted the first ever resolution on addressing the challenges of persons living with a rare disease and their families, with the hope of making a difference for the over 400 million people suffering from a rare disease globally. The resolution was adopted by all 193 UN member states and is the first UN document to recognize the specific challenges of people living with a rare disease. Here in the US, 1 in 10 Americans are impacted by a rare disease, which means that many of us here in this room either have a rare disease or know someone impacted by a rare disease, whether friends, family, neighbors, or colleagues. There are now over 10,000 identified rare diseases, and what is staggering about this statistic is that still over 95% of these diseases have no available treatment. With more than 95% of rare diseases without a treatment, I challenge us all in this room to ask the critical question, why? Why do the vast majority of patients with a rare disease continue to have no viable options for treatment of their condition? And collectively, what can we do to change this? At BridgeBio, along with other industry biotechs, we believe that we have a meaningful role to play in changing the trajectory for the rare disease community. BridgeBio Pharma is a Bay Area biotech company founded in 2015. Our goal is to discover, create, test, and deliver transformative medicines to treat patients who suffer from genetic diseases with clear genetic drivers. In 2021, just 6 years after the company was founded, we received our first 2 approvals, what is notable is that both approvals were for ultra-rare diseases. The first approval was for molybdenum cofactor deficiency type A, an ultra-rare metabolic disorder presenting in just the first few days of life, causing intractable seizures, brain injury, and death in early childhood around the age of 4. The approval was based on clinical trials that showed a meaningful increase in survival compared to the natural history of the disease. The second product was approved in the same year for another ultra-rare disease, FGFR2 fusion-driven cholangiocarcinoma, representing just 15 to 20% of the 20,000 people in the US and EU with cholangiocarcinoma. This approval was granted under the accelerated approval pathway. Now, while our unique business model and scientific approach of targeting disease at its source has enabled us to quickly develop these 2 treatments effectively and efficiently for patients, the reality is that both programs required extensive collaboration with the FDA and the ability to leverage regulatory flexibility given the rarity of the diseases. As mentioned, the first approval leveraged natural history as a comparator instead of a placebo control. The second leveraged accelerated approval to bring treatments to patients. Arguably, neither of these treatments would have been made available if the necessary regulatory flexibility was not applied. These 2 examples, unfortunately, do not happen as often as we need them to. More often than not, as already mentioned, programs are ended prematurely due to insurmountable complexities presented in the development and regulatory pathway. It is well understood that all rare diseases will require incentives and regulatory flexibilities to ensure treatments are developed. We must all remember that in the 1970s, prior to the Orphan Drug Act, there were only 10 drugs approved to treat rare diseases. 10. And while the Orphan Drug Act has spurred innovation in rare diseases more broadly, we do need to think about whether more is needed to ensure that all categories of rare diseases have a development and commercialization path. Now, this slide I've tried to illustrate at least some representation of categorization of segments of rare diseases. However, there's no clear definition of these categories of rare diseases. For the sake of discussion, I've placed them into these 3 categories. First being just more broadly encompassing everything under rare, representing people under 200,000 people. The second category in the middle, which is why we're here, ultra rare, which also includes some of the N of 1s, and depending on which reports you're reading, could range from diseases impacting 2,000 people to 10,000 people in the US. What is interesting is that reports suggest that the vast majority of rare diseases are actually considered ultra-rare, especially with advancements in precision medicine. Actually, an analysis done by the Every Life Foundation, who is sponsoring this event, found that about 85% of rare diseases occur in fewer than 5,000 US patients. And of course, we have to always remember the N of 1 diseases that impact patients in the single digits. Now that we know the rare disease complexity, we also understand that the application of existing regulatory framework can present challenges in development and are especially impactful for ultra-rare diseases. I've listed a few here that we are all too familiar with: disease heterogeneity and limited natural history understanding, lack of regulatory precedent. Many of these diseases have no available treatment, which means there's no clear and understood path forward. There's no understanding of what trial design needs to look like. There's no understanding of endpoints. There's no understanding of patient populations. There's also no clear path for novel endpoint acceptance. So when you do establish an endpoint, how do you get that endpoint accepted within the regulatory framework in order to advance your clinical development program? And quite often, we still find ourselves in conversations with regulators where randomized placebo-controlled studies are still expected. There's inconsistent use of the accelerated approval pathway, which we know for many of these genetic diseases would be very appropriate, and heightened focus on feels, functions, and survives clinical endpoints, which quite often for many diseases would result in clinical trials that would span many, many years. And at that time, given how devastating these diseases are, it's unclear how many patients would have lost their lives or progressed too far beyond where those treatments would be helpful for them. These challenges are even more apparent for ultra-rare diseases and can be devastating for programs. The result of this is the vast majority of compounds not progressing into clinical development, and certainly not progressing to the bedside of the many people waiting for them. At BridgeBio, we have a broad portfolio that spans genetic diseases of different sizes. We continue to advance development in very small populations in order to provide patients with treatment options for some of the most devastating diseases. For example, we are currently developing a treatment for limb-girdle muscular dystrophy type 2I, a genetic disease that is caused by mutations in the FKRP gene that is found throughout the body, and especially in the brain, heart, and skeletal muscles. This disease causes weakness in muscles close to the body and can also cause difficulty walking and also dilated cardiomyopathy, as well as respiratory difficulties. Additionally, we're developing a treatment for another ultra-rare disease known as Canavan disease, an ultra-rare neurological disease that affects children from birth with no treatment options beyond supportive care. As we advance both programs, we recognize that innovative clinical trial design approaches, including leveraging single trials given the very small populations, the ability to leverage novel endpoints, including biomarkers to support accelerated approval, incorporating patient experience data to help regulators with the interpretation of small data sets, and approaches to leveraging natural history are all critical to ensure development. Essentially, the regulatory tools available to the regulators must keep pace with scientific advances in order to enable treatments such as those for ultra-rare diseases to be developed quickly. With this in mind, it is imperative that Congress enable a regulatory infrastructure to accommodate the needs of such diseases. Today, I will focus on just 4 policy recommendations that I believe are critical to enabling this infrastructure. The first includes reauthorizing the Rare Pediatric Priority Review Voucher Program. This voucher, as we all probably know, is a critical incentive for developers of therapies that are being developed for rare and ultra-rare diseases, given its ability to offset R&D costs for small populations that would otherwise not be commercially viable. It is important and critically imperative that Congress pass the Creating Hope Reauthorization Act of 2024 to renew this program. The second change that I would recommend is the need to establish a framework for accelerated approval. We've all heard probably about the term primary disease activity biomarkers, which has been coined by Emil Kakkis at Ultragenics, essentially biomarkers that are at the root cause of disease, as an important option when clinical data are impossible or impractical to collect in the context of a clinical trial. New legislation implementing a primary disease activity biomarker framework for accelerated approval could greatly improve the development of therapies for rare and ultra-rare diseases. The third, establishing science-focused drug development meetings, akin to patient-focused drug development meetings, but quite often what we hear is after these PFDD meetings, the logical next step of implementing some aligned scientific approaches in clinical development programs is often still lost. With over 10,000 rare diseases, it is unreasonable for us to expect that FDA reviewers will be experts in every rare disease, or that they have even encountered a patient with the disease that they are now charged with making the most critical life-changing decisions. This increases the importance of identifying approaches to enable the co-education of FDA reviewers and developers in order to keep pace with the advances in science and ensure informed decision-making. Creating an approach to enable what I'm calling science-focused drug development meetings would allow FDA to systematically obtain scientific alignment from scientific and medical experts on drug development for rare diseases in order to inform decision-making. And the last policy change that I would recommend is strengthening the use of patient experience data. We all know that patients are the experts in their own disease. And given how rare some of these diseases are, it is critical that their experiences and the data collected directly from the patient voice be considered in the context of review documents and the data collected from clinical trials. And so we encourage Congress to build on the progress made to encourage the patient experience data into regulatory decision-making by passing the Benefit Act, which would ensure that patient experience data, patient-focused drug development, and related data are considered as part of the risk-benefit assessment that supports the approval decisions of ultra-rare diseases. Enabling these 4 policy changes, amongst others, would critically impact the trajectory of ultra-rare diseases and meaningfully impact the lives of millions of Americans. We owe it to our communities to make these changes now. I thank you for the opportunity to share these remarks and look forward to collaborating with all the stakeholders to make a difference. Thank you.
All right, thank you, Dora. Next up, Dr. Dominique Pichard. Dr. Dominique Pichard is the director of NCATS's Division of Rare Diseases Research and Innovation, where she leads rare disease research efforts. Before joining NCATS, Pichard was the chief science officer at the International Rett Syndrome Foundation. There, Dr. Pichard engaged in patient advocacy and created several initiatives to improve translational research in Rett syndrome, providing the grounds for successful drug development. Prior to her tenure at the International Rett Syndrome Foundation, Pichard was a clinical researcher at the National Cancer Institute and the National Institute of Arthritis and Musculoskeletal and Skin Diseases. In addition to her work in rare disease research, Pichard is mother to a daughter who lives with Rett syndrome, so she has a deep understanding of the many challenges associated with having a rare disease. She is passionate about improving the system for all those affected by rare diseases, which aligns with NCATS's mission of bringing more treatments to all people more quickly. Pichard received her MD from Georgetown University School of Medicine and her MS from the University of Maryland School of Medicine.
All right.
Thank you very much, Ethan. And thank you for allowing me to be here to speak with you this afternoon. As Ethan mentioned, I'm Dominique Pichard, and I'm the Director of the Division of Rare Diseases Research Innovation at the National Center for Advancing Translational Sciences, or NCATS, at the NIH. We've already spoken about this several times today, and so I really don't have to dwell on this. The challenges with rare diseases. There are more than 10,000 known rare diseases based on science and technology today. That number will continue to increase. And as you've heard already today, only about 5% of those rare diseases have treatments. There's a tremendous gap in getting us to every individual affected by rare diseases with treatments to change the course of their disease, to give them an improved opportunity for life without their disease. The other challenge that we haven't really talked about today is the time that it actually takes. So once you make that discovery in your basic science lab, the black box from getting there to your FDA-approved treatments. And that is really where NCATS spends a lot of its time, in that translational science. We need to make these discoveries actually into treatments, and that is a tremendous period of time. You see, on average, it takes, with our historical data of kind of the traditional drug development, 10 to 15 years from that discovery until it's in your medicine cabinet or the infusion in the hospital that you're able to get. And 9 out of 10 of those clinical trials, promising therapeutic candidates, will fail. Huge failure rate, tremendously long development timeline. 95% of rare diseases don't have treatments. So the NCATS mission is really to turn these research observations into health solutions through translational sciences. How can we improve the process? And as was mentioned, the vision at NCATS is to have more treatments for all people more quickly. And really we work to re-engineer that translational pipeline so that there are roadblocks that we know about within the translational pipeline, that 10 to 15 year time development. How can we smooth out those roadblocks, create efficiencies, find commonalities so that we can get to our end goal of more treatments more quickly? And what is our approach? So really, these are kind of some highlights of our approaches, but really importantly is that first area, understanding what's similar across diseases. With more than 10,000 rare diseases, if we go one disease at a time and we reinvent the wheel each time, we all do the same thing. For every single one of the diseases, we have to understand the disease. We have to get a detailed natural history. We have to develop clinical trial endpoints, so these outcome measures or biomarkers that was just being talked about for every single one of these diseases. That's not even talking about developing those therapeutics. So that's not talking about each individual drug, what has to go into, or gene therapies. And so we really want to understand what is similar not only across diseases, but across the mechanisms, the processes, so that we can create efficiencies. We want to develop models that better predict a person's reactions to treatment so that we can lower that failure rate. We want to enhance clinical trials so that the results more accurately reflect the patient population. And growing more and more, we see the need to leverage real-world data and data science approaches to address the public health need. Rare diseases really have a home at NCATS. I think rare diseases exemplify a lot of the translational science challenges and issues. And by approaching those roadblocks in rare disease research, there's a much broader impact in translational science. And so NCATS really develops new ways to understand and treat rare diseases. And we do this in, I'll say 3 broad buckets. We create innovative opportunities that provide funding to advance medical research. We try to use data-driven solutions. And again, this will be increasing as more data is available. When you have rare diseases and there's not much data available, it's challenging to have data-driven solutions. So we need to create these data-driven solutions, create the opportunities to have them. Really importantly is the resources for our patient and patient advocacy community. You've already heard this afternoon, patients are part of the solution. Patients, their caregivers, their advocates, they are sometimes the ones creating the solutions. We need to make sure that we are involving them in the research process early. So at NCATS, we spend quite a bit of our efforts working with the patient community, creating opportunities to educate them, to empower them, so that then when they are moving forward with building foundations, raising funds, that they know what to do and how to do it, so that we can all move forward together. I'm going to tell you about a few of our initiatives that really exemplify some of the work that I've been talking about and what our approach is. So I'm going to start with the Rare Diseases Clinical Research Network. This was established in 2002, in the Rare Disease Act of 2002. And this program was initiated in 2003, and it's recompeted every 5 years. So we are currently actually have a funding opportunity out to initiate the next cycle. What this did is it allowed us to create this research network of individual consortium for rare diseases. And this next slide really highlights our current network. Each one of these is a different consortium that has to include at least 3 rare diseases. So again, we're going more than one disease at a time. We need to find the commonalities. As you can see on the left-hand side, the key stakeholders are the NIH, and along the bottom are the different institutes that work along with the National Center for Advancing Translational Sciences. So we fund these opportunities. There are the researchers and the clinicians who have applied and are running these research teams. Every single consortium has to engage the patients and the patient advocates group. They are part of that research team. And that is really important because, as you've heard, the patients have to drive the solutions. They know what is most important. They know how to improve that field function. Survival is a little bit more clear. But the field and function, that has to be driven from the patients. They can guide what's important in this research. And so each individual consortium, as I mentioned, has 3+ diseases. But in this network, I've seen it. I've been at NCATS for about 9 months so far, and I've seen this group engage. They are learning from each other. What one group develops as an outcome measure and presents it to the group, the next group says, we have similar issues in our disorders, our group of disorders. So now we're amplifying. The work that is funded for one group really can have a much broader impact. And really what we want is to get treatments. And this slide highlights the translational impact in terms of actually getting treatments to patients. So we have studied across the years, across the cycles. There have been about 200 rare diseases that have been studied in the RDCRN. We have developed the infrastructure. So they all have a natural history study. They usually work on some other clinical trial outcome assessment development or biomarker assessment, which really then lays the groundwork to be able to get commercial interest for developing therapeutics. And what this slide is showing you, the 12 FDA-approved treatments for 11 of these diseases. And it's not to say that these were pivotal studies funded by the RDCRN, but rather RDCRN did all the hard work, allowed these diseases to be ones that could be of commercial interest, and then pharmaceutical companies brought this across the finish line. Now, while 12 does not seem like a very large number when we're talking about more than 10,000 rare diseases, I think I'm very encouraged by this when you see the dates of the approvals. 5 of them have been in the last year. And that also goes to show why this process, it really demonstrates how this process can be quite slow. You have to do all that groundwork. It can take quite a bit of time to understand the diseases well enough to develop those outcome measures so that you can have successful clinical trials. And by setting up this infrastructure in it, and I will say that about 70 to 80% of rare diseases have a genetic mutation, a genetic origin. And so this is really for the majority of rare diseases. So you have a gene that's faulty. You want to replace that faulty gene with a correct gene. And so through this AAV gene therapy, you use this viral vector to deliver the correct gene to the cells of the body where they need to go. And then, we'll call this delivery box, gives you the corrected gene, and your body does what it needs to do to make this into the corrected protein. So really oversimplified. But what it's doing is getting to the root of the issue. And so this really is something that for the genetic diseases is of high priority. So two programs that really look at this gene therapy are the Platform Vector Gene Therapy Program. And so this was really meant to say, how can we create a system that will ultimately be a plug and play for all disorders that come down the road? And the idea was, let's lay out the framework. Let's solve many of the questions that are associated with gene therapy. It is quite complex to do the manufacturing because your little delivery box is actually different. So this is one of the milestones was the PGT successfully navigated the Food and Drug Administration orphan drug and rare pediatric disease designations for this particular gene therapy. So there are several candidate gene therapy programs through this. And so each one, we're learning each time we go, and we'll be sharing the learnings so that the pharmaceutical companies or patient advocacy groups that are leading these efforts can learn from our successes and our mistakes. Next, just to touch on another gene therapy program called the Bespoke Gene Therapy Consortium. This is a public-private partnership, and the goals of this are, again, to make this AAV technology more accessible by optimizing this production and sharing the learnings of this. So not only are we optimizing it for the particular diseases that are being tested in this consortium, but sharing the learnings. We want to streamline all of that work, that 10 to 15 year process, so that the preclinical and product testing is really much more streamlined. I will say gene therapy is relatively new kid on the block compared to small molecules, and so there's a lot still to be learned about the process for the early phase testing, that preclinical, as well as the product testing. This next slide shows the public-private partners that have been engaged and the commitments. And so this has really been a huge lift and tremendous partnerships that have allowed this program over the last almost 2 years now to continue to grow, and hopefully we'll soon be talking about the clinical trials for these diseases. I'll actually skip through. So this is the portfolio of the diseases that were selected. This was an application process where researchers and patient advocates together could apply to be selected as a disease. All of these diseases had to be ones of no commercial interest. That was the goal. We want to do this to show how we can take diseases, really small populations, really don't have that infrastructure yet, are of no commercial interest, and still have successful gene therapy trials so that the next ones that come along can follow the playbook. And in fact, we have released a playbook. This was the version 1.0, which was released last month at the Accelerating Medicines Partnership annual meeting. And this is a playbook that helps the research groups and the companies in gene therapy just understand the process. It just simplifies it and tells them kind of how to put one foot in front of the other for this gene therapy. And as we continue along this pathway, we will iterate and create new versions of this playbook with new learnings. And finally, I want to talk about one last program, somatic cell genome editing. So gene therapy is really exciting, and it is the area of genetic medicine that has been around longer. And so there's quite a few programs that are coming along, but the next set of technology that's exciting is gene editing. Again, this would be useful for the 70 to 80% of rare diseases that are of genetic origin. And so this somatic cell genome editing program is intended to develop novel technologies to build that foundation for clinical application, and then deliver the resources to accelerate treatments. So in phase 2, we are actually looking to find better ways to actually get those editors to really challenging organs such as the brain. And so there have been these challenges, these prize challenges, where we seek to get the most innovative solutions for this significant challenge of genome editing. How do you develop this technology, and how do you deliver it successfully to the organs that need to be delivered? And you know, I think in all of these, I'm going to skip ahead to this one, the underlying goal in NCATS is to, as I said, our vision is to get to more treatments more quickly for all people. And the only way we're going to do that is if we work many diseases at a time. So we need to find those commonalities. We need to see where do diseases have overlap, either in their molecular etiologies, in the approach to their treatment, or in the symptoms. How can we look across the diseases, find those commonalities? And how do we develop therapeutic platforms, such as this bespoke gene therapy or the somatic cell genome editing, that can ultimately get us to those treatments faster? Thank you.
All right, last but certainly not least, Annie Kennedy has served within the community for nearly 3 decades through her roles with the EveryLife Foundation, Parent Project Muscular Dystrophy, and the Muscular Dystrophy Association. At that time, she helped lead legislative efforts around passage and implementation of the MD CARE Act, the Patient-Focused Impact Assessment Act, the Newborn Screening Saves Lives Act, and key provisions within PDUFA 6 and the 21st Century Cures Act. She has been integral in the numerous rare communities, helping lead newborn screening programs, regulatory engagement, and access efforts, including engagement with ICER around product-specific assessments, as well as the development of the modified framework for the evaluation of ultra-rare diseases. Since joining EveryLife in 2018, Annie's leadership has included the National Economic Burden of Rare Disease Study, the ICD Code Roadmap, the development of the Guide to Patient Involvement in Rare Disease Therapy Involvement, and the Cost of Delayed Diagnosis in Rare Diseases, a Health Economic Study. Annie, take it away.
Thank you. Thanks, everyone. So everybody up here has done such a phenomenal job that a lot of what I was going to do has already been done. So I'm going to actually do this fairly swiftly so that I could maybe yield a few minutes back so that Maggie can have her moment that she really deserves, and I'd really like to see the end of that video. I don't know about everyone else. So we've been talking around this, but one of the reasons why this briefing is really important today is that there is no statutory definition for ultra-rare in the U.S. And we maybe should have started with that for those that don't know that. But there are a number of definitions that are floating about and are used in different contexts in different ways. And there are just a few examples of those up here. And we do have entities in the U.S. that have developed definitions that are being utilized, but there is no definition here in the U.S. And there are a lot of reasons for that, which we won't go into. But I will do a little commercial for the fact that there is a scientific workshop being convened tomorrow by EveryLife, where scientific and regulatory leaders are coming together to talk about the potential for the consequences of developing a definition, and whether or not that would actually be a numeric threshold or a characterization, and how that could potentially be beneficial. That said, in advance of that scientific workshop, we disseminated a survey to the patient advocacy organization leaders that would be attending that workshop. So these are stakeholders who are super invested in ultra-rare, such as Ethan and Christine, who are sitting here beside me, who are leading organizations and driving development in the ultra-rare space themselves. And we asked them to complete a survey around their perceptions and their characterization of ultra-rare. And this is just a snapshot of some of the input that we received, just to show that even among those expert stakeholders, there are a lot of differences of opinion around where that threshold might be. And this is just a quick snapshot of that numeric threshold. So as a community, it's really important that we do have a scientific discussion around this, and to better understand how that threshold would be used and what the potential implementation of that could look like. So this presentation is really about what we need in order to be able to solve for ultra-rare. How do we make sure that we change outcomes for individuals living with ultra-rare diseases in the U.S.? And so we really are categorizing that in a few different, we're talking a lot about buckets up here. And so we can call them buckets, we can call them puzzle pieces. But how do we ensure that there are more incentives for developing therapies in the ultra-rare space? Christine really gave a very striking example of what happens when there aren't more incentives. And so just to really make it clear, part of the difficulty and challenge in ultra-rare is that the incentive for a company to develop a product in ultra-rare is not the same as the incentive for if you're doing male pattern hair loss or arthritis. The market share just isn't there. And so we really have to think about what are the incentives? How do we de-risk that space for an investor or for a company to work in that space? And so these incentives are critically important. Resources. We have to make sure that the stakeholders who are working in these spaces are resourced to do this work. And the question is, do we need to resource them additionally above and beyond how others working in the space are resourced? And that is a really important policy area for us to be working in. And that really leads to the ultra-rare policies. And those working in ultra-rare are disadvantaged above and beyond those in even the more broader rares and those certainly in the more chronic spaces. And then infrastructure. Infrastructure is difficult across the board, and especially when we think about ultra-rare. And so just a couple examples, and these are certainly not going to be an exhaustive list, but my colleague, Dr. Ndu, already talked about the Creating Hope Reauthorization Act, and she did a really beautiful job talking about this, so I'm not going to belabor it. But the Pediatric Priority Review Voucher has been an incredibly important incentive, so in that incentive category for developing products in rare diseases, and this is an incredibly important incentive in the ultra-rare space. We cannot afford for this incentive to go away. There are already too few incentives in rare, and we must ensure that this gets reauthorized. We are very close. This was a part of that markup last Thursday, and we need to make sure that we get this one over the finish line. Another category that we talk about are resources, and one way that we work on those resources here on the Hill is through appropriations. These are three categories that we have been working on as a rare disease community around appropriations and making sure that our federal agency partners are resourced. But just a couple examples around how this becomes really incredibly complex and incredibly important when we think about ultra-rare. Christine talked about newborn screening. So newborn screening is an area that we know to be life-saving in rare disease, but we also know this is a public health program. And so when the Secretary's Advisory Committee of Heritable Disorders in Newborns and Children thinks about the benefit and risk of adding a condition to that screen, they think about the cost of adding a screen. And so what will happen when it's an ultra-rare condition that they're considering be added, because they're now going to have to screen a whole population for an ultra-rare condition. EveryLife actually offered testimony last week, as did many others in the rare community, to say that we argue for the fact that we must ensure that we are working to save the life of every baby in the nation, regardless of how often that condition occurs. But that is something that we have to say is a moral imperative, regardless of incidence and prevalence, that we continue to fight for. But that comes up in the context of public health. Translational research. So this is another category. We just heard a really important presentation on what NCATS is working on. We have to make sure that NCATS and then FDA, and there's another section of this slide that talks about the orphan products grants programs, are resourced adequately and then additionally incentivized to make sure that as those funds and those projects are being prioritized, that small populations and ultra-rare populations are being able to be focused on and worked on. This is a priority area when we think about policy areas. So we have just a couple things we'll flag on policy areas. ICD codes. So if you're in this room and you're a staffer, you may not know what an ICD code is, but an ICD code is your International Classification of Disease. I like to think of it as your hashtag that follows you throughout all of your medical records. So it's high blood pressure, it's pregnancy, it's sutures. You have a hashtag for any medical encounter that you have. Out of the more than 10,000 rare diseases, fewer than 600 of them have an ICD code, which means that rare diseases are essentially invisible in the healthcare system. And this is a barrier from a multitude of reasons, and it is one that is surmountable. We can fix this, but we actually need to develop policies that help us do that. A couple other policy issues. Christine talked about, at the very beginning of her presentation, she did a beautiful job talking about why off-label products are so important in the GM1 population, because so few rare diseases have approved products for that specific rare disease. So many patients take products that have not been approved specifically for their rare disease, but can treat the symptoms associated with rare disease. They're very difficult, though, to get reimbursed when they're off-label. There is a bill here in Congress, the Protect Rare Act, that would work to address that. So policies that look to help overcome hurdles to access that specifically address issues that would target our ultra-rare community are incredibly important. And then Dr. Ndu talked about the accelerated approval pathway, and she talked about PDABs, which are incredibly important. I would also just highlight that in the end of 2022, FDORA, or the Food and Drug Omnibus Reauthorization Act, included enhancements to the accelerated approval pathway that were incredibly important. But one of them is one that I would just like to highlight because it became a little bit of a barrier to ultra-rare. So when we solve for other problems, sometimes we create problems elsewhere. One of them was that the FDA is now permitted to require that a confirmatory trial be underway at the time that a product is approved. So that was something that happened during FDORA. If you are a company that has never had a product approved before, which often happens in small biotechs, and you have an accelerated approval product that is now moving through the regulatory space based on a surrogate, so that's what's happening in accelerated approval, and you're now going to potentially be required by FDA to get ready to stand up your phase 4, your confirmatory trials, before you know if FDA has approved that product, before you know if FDA has decided that your surrogate is confirmed, you're asking small biotechs that don't have a financial runway to invest in post-market studies. But the other thing that happens, so that's a problem for all rare companies, but especially in ultra-rare, that company has probably exhausted the whole US population and the ultra-rare population for their studies. So you're probably asking those companies to have to go outside the US to design those trials and recruit for those trials. They're going to have to set up new trial sites and recruit new patients. We're asking companies to do something that is almost impossible and definitely beyond their affordability. So we need to think about the ultra-rare community when we think about developing these policies. Okay, and then the last bucket is infrastructure. And we have been, as a rare community, moving forward proposals to develop infrastructure that works better for the rare disease community and just makes sense. And we're not just making this up. We're looking at models that work in other communities. So one of that is the FDA Rare Disease Center of Excellence. And FDA already has the congressional authority to develop centers of excellence, and we've seen this work beautifully in oncology, and we would like to see that work in rare disease. The other is we have seen interagency coordinating committees work beautifully. So coordinating committees where we bring together NIH, FDA, CMS, the Social Security Administration, HRSA, to work together to share what their agendas in rare are, and then develop shared agendas to knock down obstacles and increase momentum in rare disease. And so that is a separate request that we are asking for the establishment of an interagency coordinating committee in rare diseases. So thank you so much. There is so much that we can do even before we have determined whether and what an ultra-rare definition would be. And then so much more we will be able to do after we have come together to discuss that. And that discussion is happening tomorrow. Please look for the link. Thank you. I think you should come Maggie up. Set a question.
All right, by popular demand, let's see if I can get us back. Oh boy, let's see, how do I just jump back to the beginning? And we're going to do a round of questions in a moment, so stay tuned, but thank you for indulging me. Let me play the rest of this video for you. Oop, geez. Okay, as promised. Okay, maybe while this is going, since you've seen this part, I can kind of maybe tell you a bit more about the backstory. So I actually met Maggie's mom, Holly, through a guy named Matt Might. Maybe some folks here know this name, Dr. Matt Might. His son was one of the first children ever to get a diagnostic whole exome sequence in like 2010. And there was a New Yorker written about how his child was the first with that disease. In any case, we got connected when Holly was on a tour of the White House because Matt was working at the Precision Medicine Initiative, I think, at the end of the Obama administration. So actually, we got together because of DC at the end of the day. But this is her baseline. Remember, this is the Grand Collar Hotel and Mayo Clinic. And this was about 4 months before she started taking the drug. And I should maybe comment on how we found this drug. So this drug is called apalirostat. It was a drug approved in Japan in 1992 for the treatment of diabetic neuropathy in adults. And we found, using very fast and efficient disease models, specifically yeast and worms, that we could recreate the disease in those simple critters. And then we discovered this drug, that it had this effect. So here's 3 months after. I asked everybody if they noticed a difference to raise their hand. She's executing a really nice sharp left turn here into the elevator. Turn at the end. And this is a nice straightaway to give you a sense of her gait and how she's able to get herself moving. Oh, didn't even notice this cheesy music. Let me turn that off. So this is 6 months on the medication. And then we'll play another clip, I think, from Mayo Clinic. This maybe will be in October of 2020. So as the world was collapsing in January 2020, Maggie was taking her first dose and was starting a very different trajectory. Okay, just bear with me here. And of course, I want to give a shout out to the principal investigator, Dr. Eva Morava Kozic, who was running this trial. She's been Maggie's doctor from the beginning. She's the queen of CDG. She's one of the world's experts in this kind of inherited metabolic disease. So Maggie's still scooting around here. She's able to navigate these turns and obstacles. So remember, she started on January of 2020. Then I think we'll skip ahead. We'll zoom ahead here to an outdoor scene, I think. So obviously, this is not a cure, but I think the message here is that drug repurposing holds great promise for you to be able to take something off the shelf and stabilize the situation, or at least prepare these kiddos for the more experimental treatments that are more targeted, and they start with a hopefully more robust foundation. This is Maggie strutting her stuff outside, and then here we go, the coup de grace. So this is almost 2 years on the drug, and with some support here from one of her aides, she's able to do a nice runway walk. And I should say that, obviously, this N of 1 inspired an N of 2. I showed you that, those clips. And then those 2 studies inspired the community. And now we have a 38-patient pivotal phase 3 study, fully enrolled. It's still in the randomized phase. I can't tell you much more. But we have data readouts coming later this year. And we're very excited for what's next. In fact, we've already found a second-generation repurposed drug that we want to add on top of that. Thank you, everybody. Okay, so with that, why don't we go on to questions? I think we have sufficient time for questions.
Just maybe one.
Okay, we'll do the lightning round question. Okay, great. All right, so, where's my slide? There we go. So this is a lightning round question. This is going to be a question for everyone on the panel. Everyone will get a chance to answer this. So our hope is that today's briefing shines a light on the small, rare disease patient populations and the challenges in developing treatments for these communities. What is the one thing you hope that viewers and attendees take away from this briefing? Let's start from the top with Christine.
I really hope that you see the need for change and the support for ultra-rare diseases.
I would echo the same.
I would echo the same. We really need to think about drug development in this space very differently. We need to think about our regulatory flexibilities differently as well in order to ensure that treatments get to patients.
I hope that everyone here walks away remembering, if we don't move more than one disease or more than one process and platform at a time, that it's going to be not in any of our lifetimes that we see treatments for even the majority of the rare diseases that we currently know about.
I would just hope that Maggie and Iris and the families that you've met are what stick in your head today, if you remember nothing else, other than there is something we can do today. There are policies that will impact the families that we've met today, and that time is a commodity. So that the families today who are living with ultra-rare diseases don't have time, and that we need to lean in today.
Questions?
Do you want to answer it too?
Yeah, I guess I think people have talked a lot about how the word rare in rare diseases is not great marketing. And so I've been starting to talk about things like the invisible war, the plague of rare diseases. I think we need to be a little bit more dramatic because we're not really connecting, because it's easy to hear rare, 10,000 plus, and you're just like, your eyes gloss over, and it can only be contextualized if you see actual human beings. But maybe if we start, I don't like war, but maybe we need to acknowledge, though, that this is a vicious enemy. And this is not, and if you do nothing, inaction has tremendous costs. So you have to have a warlike stance. But hopefully, I think if we start using language that really activates people and mobilizes people, I don't have a better substitute for rare diseases, but I think talking less about the object and more about the action that we're taking might help mobilize more people.
Wonderful. All right. Thank you so much, everyone, for being here today, and thank you, Ethan, for being a wonderful moderator. We appreciate everyone today.
What's up, Christine?
So good to see you, Lucy.
Are there any other companies that are coming out to pick them up?
I mean, Castle Bio.